I decided that we needed a short video, short notes video about gene linkage. This is an important topic, and you'll encounter some problems on your test this week and also on AP exam that involve gene linkage, and so wanted to make sure that you had a little bit of instruction about how to handle those kind of problems and things like that you might not get just from reading and taking notes on your own. So big thing to realize is that we know what we know about gene linkage or about how genes are organized on chromosomes thanks to the work of Thomas Hunt Morgan a little over 100 years ago. Okay, he worked with Drosophila melanogaster, which is a fruit fly, and it's become a very popular organism to use in genetic research. Big thing is they produce a lot of offspring. You can have a whole new generation in 2 weeks, and they only have 8 chromosomes, 2 pairs of chromosomes, so it's pretty easy to figure stuff out. And there are various mutant phenotypes that you can look at and do test crosses really easily to see what they have. I personally don't like working with fruit flies, but their genetics is a really important thing to understand, and they help us understand a lot about how things work. So one of the first things he noticed is that there were various kinds of phenotypes that were common in fly populations. Okay, the ones that are most common are called the wild type or normal. Try to use the term wild type instead of normal because of the negative connotations there. But the ones that are opposite of that or different than that are called mutant phenotypes. And so one of the first ones that he discovered was the eye color. Most of the fruit flies had red eyes, but occasionally there were some that had white eyes. And so he first set out to investigate how that eye color was inherited. So he mated male flies with white eyes with female flies with red eyes. Okay, and in the F1 generation, the first generation that resulted from that, all had red eyes. So okay, obviously red eyes are dominant. Then when he allowed the F1s to mate together, they produced an F2 generation that showed a classical 3-to-1 red to white ratio that you would expect for a dominant recessive trait, except that the only ones with white eyes were males. No females had white eyes. So that was really interesting, okay? So he figured that maybe the eye color was related to the sex of the fly. So he had to look at that. All right. So the big thing is that the inheritance of sex in fruit flies is similar to what it is in humans, in that we have X chromosomes and Y chromosomes. Females have 2 X chromosomes, males have an X and a Y. The chromosomes are vastly different, of course, in fruit flies and humans, but it's the same type of sex determination. And so what he did was he did this cross, and he found that all the ones that had white eyes here, so he figured out that perhaps that if that gene for white eyes was only on the X chromosome and not present on the Y, then if the offspring got the mutant allele, then they would end up with white eyes only if they were male, because they would always, if they were female, they would have the dominant wild type red eye allele. He didn't call them alleles at the time, but that's what we call them now, so you understand how that works. So that was the first thing. He did lots of different things with fruit flies to show basically that there are lots of different things that can be linked, okay? And so in all of his research with flies, he came up with lots and lots of different kinds of phenotypes of different kinds of traits that he could look at and determine, what chromosome they were on and so forth. So he crossed flies, first of all, that differed in traits of body color and wing size. So he's looking at 2 different traits at the same time. All right, he found that body color and wing size are usually inherited together in specific combinations. These are called parental phenotypes. And so he said, well, okay, since they don't seem to assort independently, they must be on the same chromosome. And that's a pretty logical jump to make when you understand that genes are found on chromosomes. So if they're both on the same chromosome, then you would tend to inherit them together. VG stands for vestigial, and so those are basically small wings. And there are some other traits related to wings as well, but this particular one that he's talking about is the body color and the size of the wings, whether they are normal size or whether they are vestigial, okay? And so here we have the first cross that we have here. So what the wild type has a gray body and normal wings, and then we have a double mutant here that has a black body and vestigial wings. And so when he crossed those, he found that you would expect if they're inherited together that the dihybrid is going to have gray body and normal wings, and that the homozygous recessive is going to have a black body and vestigial wings. Pretty logical jump to make, okay? But here's what he found, that also found some that were not the same phenotypes as the parents. We call those non-parental phenotypes. And so that is the first clue that anybody had about crossing over, which we understand now is something that happens in meiosis. But this is how he figured out, or how it was figured out, that there was crossing over that occurred during the formation of the gametes. All right. So if here are the numbers he came up with. Okay, you would expect if the genes were on different chromosomes, you would expect a 1-to-1 to 1-to-1 ratio of all of those things. And if they're on the same chromosome, you would expect to have more of the, or some always of the parent genotype or phenotype, and none of the non-parental phenotype. What he found was that he had most of them had the parental phenotype, but there were some that did not have that. So there was some kind of recombination going on there. I'm just going to give you a big hint right now. Whenever you see numbers that look like this, where you see large numbers of the parental phenotypes and much smaller numbers of the non-parental phenotypes, that is an indication that those alleles are on the same chromosome and that crossing over has occurred. But they're pretty close. That means they're pretty close together. It doesn't happen very often, but it does happen from time to time. That is a big hint. So when you see numbers like that in problems, you need to think about the linkage issue. Okay. So he did various kinds of crosses, test crosses to see. Okay, here's one again showing the wild type with a normal body, with a gray body and normal size wings, with the double mutant there. Okay, and there's our dihybrid, our F1. Okay, and then he, instead of crossing the F1s together, he took one of those F1 dihybrids and crossed it again with the parental phenotype that was the male with the black body and vestigial wings. So that male could only produce that one kind of chromosome, because even if crossing over happened, it's the same allele because they're homozygous recessive. Okay, and so what he found was that in the female, sometimes you got the recombinant chromosomes as well, and that ended up with different results from your offspring. Okay, so here he had the parental type offspring. Okay, and then we had the recombinant offspring, and so he was able to calculate something called a recombination frequency. Now you can do this, you can figure out how often that changes over, okay, how often the crossing over occurs, and that's an indication of how close together or far apart the genes are on the same chromosome. Alfred Sturtevant, who was one of Morgan's grad students, constructed a map of the genes, okay, and listed the loci, the locations of genes on a particular chromosome. And he predicted that the farther apart the genes are, the higher the probability that crossover will occur, and therefore the higher the recombination frequency. And so using those percentages, those recombination frequencies, he was able to develop a gene map. Here's a linkage map, and this shows one of the things. This is a couple of different things. Black body, I think, and cinnabar eyes and vestigial wings, I think, are the ones that those stand for. It's really irrelevant. The point is that you can figure out the distance between genes by calculating that recombination frequency. And basically they're expressed as something called map units. And the map units may differ in size from one species to another, but basically it indicates a 1% recombination frequency. And so these two are 17% apart, the black and the vestigial here, and between the black and the cinnabar only about 9% recombination occurs there. Between cinnabar and vestigial, about 9.5%, so that's around 17, 17 to 18, so that's pretty close. It's not a guarantee, it's not really a measurement as such, it's just it's a frequency of how frequently that happens. And so he used the recombination frequencies to make linkage maps on these fruit fly genes. And so you can also see that in lots of different things. I am not going to ask you to do that in class. However, there are sometimes questions that show up on the AP exam. They will ask you to figure out how close together, how far apart they are. I don't think it's as frequent anymore as it used to be. I wouldn't worry a whole lot about it, but this just gives you an idea of how that can be used. And that's all. I'm going to give you some practice problems to work on, just to have some practice doing various kinds of genetic crosses with linkages of various kinds. Some sex-linked, some linked on non-sex chromosomes, on autosomal chromosomes, just for practice, so you'll have an opportunity to work with those.